Assalamualaikum and good morning. My name is Siti Nur Sakina from AS 257. Today, I will present my FYP proposal on the antimicrobial effect of apple cider vinegar against selected pathogen associated with atopic dermatitis. This presentation will include the research background, literature review, and research methodology. Atopic dermatitis, also known as eczema, is a skin condition that causes dry skin, itchiness, and scaly patches. Microbiome imbalance has been identified as one of the causes of atopic dermatitis. Several microorganisms such as Staphylococcus aureus and Candida albicans have been identified to cause secondary infection in people with atopic dermatitis. The skin of atopic dermatitis patient has a higher pH and is more likely to develop infections caused by bacteria as the optimum pH of bacterial growth is close to neutral. Due to antibiotic resistance of the microbial pathogen, apple cider vinegar become more popular. This is because apple cider vinegar contains 5% acetic acid, which has strong antimicrobial properties. Thus, the low pH of apple cider vinegar has the potential to restore the skin pH as the healthy skin pH is 4.7 to 6. Problem statement. Antibiotic resistance becomes one of the biggest challenges in treating any infectious disease caused by bacteria. Thus, home remedies such as apple cider vinegar have become one of the potential treatments for atopic dermatitis. However, apple cider vinegar requires proper dilution to avoid potential side effects while treating the infection due to its acidic properties. Thus, a study is needed to determine the lowest concentration of apple cider vinegar that can inhibit bacterial and fungal growth in the nutrient agar while determining the antimicrobial activity of the apple cider vinegar. This proposal includes two research questions, which are what is the lowest concentration of apple cider vinegar that can inhibit the growth of S. aureus and C. albicans, and what are the antimicrobial effects of apple cider vinegar, bactericidal or bacteriostatic, for skin bacterial pathogen associated with atopic dermatitis. The objective of this study are to determine the lowest concentration of apple cider vinegar that can be used to inhibit the growth of S. aureus and C. albicans, and to determine the bactericidal and bacteriostatic effects of apple cider vinegar against S. aureus and C. albicans, a common pathogen for atopic dermatitis. The next part of this presentation is the literature review. First, I will explain about the pathophysiology of atopic dermatitis. Several factors have been discovered as the main causes of atopic dermatitis. The first factor is genetic, which is known as FLG null mutation. The FLG gene is responsible for encoding the production of filaggrin protein. The breakdown of the filaggrin protein produces hygroscopic amino acids that play important role in SC hydration and the acidic pH of the skin. The next factor is epidermal dysfunction. The imperfection in the proteins constituting the epidermal barrier promote irregular immune responses to antigens. The skin of atopic dermatitis patients tend to overproduce the Th2 cytokines. This cytokine cause inhibition of the antimicrobial peptides AMP. The third factor is lipids. The lamellar bodies located between corneal side of the stratum corneum are made up of a lipid matrix that is important in forming a tough structure for the skin barrier. However, the overproduction of Th2 cytokines decrease the level of lipid production. The last factor is microbes. Patients suffering from atopic dermatitis lack bacterial diversity with increase in Staphylococcus concentration. S. aureus causes upregulation of cytokines and stimulate mast cell degranulation, causing skin inflammation. There are two previous studies of the effects of apple cider vinegar against pathogens associated with eczema. The first study from Lu et al. 2021 shows that there is no significant difference in the concentration of S. aureus on the skin of the test subjects. However, the diversity of the skin microbiome of the test subjects become almost similar to the healthy subjects. This shows that apple cider vinegar has the potential to treat the lack of diversity in normal microflora. as the lack of diversity of normal microbiome is one of the causes of atopic dermatitis. Next is a study by Yanick Sharafin and Shah 2018. This study includes different concentrations of apple cider vinegar. The result after the treatment with apple cider vinegar shows that several enzymes failed to be produced by S. aureus, and the C. albicans lacks enzymes required for glycolysis after the treatment. The bacteriostatic and bactericidal effects of antimicrobial agents. Bactericidal antibiotics are responsible for killing the bacteria cell, meanwhile bacteriostatic antibiotics suppress the bacterial growth. Clinically, bactericidal is preferred as compared to bacteriostatic antibiotics, as bactericidal antibiotics can limit the development of bacterial resistance towards the antibiotic. However, some bactericidal agents are concentration dependent. Experimental design. The first step for both microbes is the isolation and identification of S. aureus and C. albicans. The identification tests for S. aureus are isolation on MSA, Gram staining, catalase test, and coagulase test. The identification method of C. albicans is microscopy identification of germ tube test. Next is the preparation of both microbes inoculum by using tryptic soy agar. The next step is the preparation of different dilutions of apple cider vinegar. The first objective will be tested by using the minimum inhibitory concentration MIC test to find the minimum concentration of apple cider vinegar that can be used to inhibit the growth of the microbes. Next is the minimum bactericidal concentrations MBC test to detect the minimum concentration of apple cider vinegar that is capable of killing the microbes. ANOVA will be used for statistical analysis. The second objective will be evaluated by using the ratio of MBC per MIC. Here is my Gantt chart for this final year project FYP. This is several references that were used in writing this final year project. That's all from me. Thank you.